This, of course, is the iconic double helix structure of DNA. It's extraordinarily beautiful as molecular structures go, even more so when you get some biological insight into how this structure makes it possible for it to do its job, which is to function as the sort of repository of genetic information in the cell. But now that you know some chemistry, the question I want us to ask here is the why question, so that we're moving on from just admiring the structure and we're asking why should these molecules fold up into this very specific arrangement. Now, of course, when DNA is made in the cell, there's a lot of heavy machinery around, enzymes and so on, which you might think would play a role in that. In other words, you might think that the reason the molecule looks like this is that the enzymes that make it kind of twist it up into that shape and it just kind of sticks there. But in fact, if you make two complementary strands of DNA in the lab so that they've never seen a living cell, they're just made by chemical synthesis, and you bring them together, they will fold up around one another to make the double helical structure. So it doesn't need any complex biological mechanisms to put the structure in place. It will self-assemble to do this. And that means that we can think of this as a chemical problem. So that's really what this is going to be about, is to use chemistry that you've learnt to see how far we can go in understanding why this structure should look the way it does. Now you can see from the colouring here that the DNA is actually made of two completely separate molecules. There are no covalent bonds between them. The only connections between them are intermolecular forces. And so a lot of this is going to be about understanding how those intermolecular forces come together to make this a feasible structure. And of course, in order to do that, we need more resolution. We need to see what the actual atoms are in the molecule. So in order to do that, we can have a look now at a so in order to do that, we can have a look now at a skeletal formula of just one of these strands to see what the actual functional groups are that we have available for forming these intermolecular forces. Okay, so there it is. Now what you can see is that DNA is a polymer, and running along this edge of it, you can see the backbone of the polymer where we get this regular repeating structure. So what you can see is that we have these 5-membered rings containing an oxygen and then a phosphate group, phosphorus with 4 oxygens around it, and another 5-membered ring, another phosphate group, and this keeps on repeating all the way through the polymer molecule. So this is a very small section with just 4 repeat units of the polymer. DNA molecules can be huge. So chromosome 1 in each of your cells, for example, has something around 260 million repeats in it. It's absolutely enormous molecule. And this is just a tiny snapshot of a very little bit of it. So this is the backbone. And these 5-membered rings are actually derived from a sugar called deoxyribose. Now, if you study biology, you probably are used to looking at sugars, and you know that they typically have these rings containing an oxygen atom like this, but that typically they're decorated with lots of OH groups. And you can see this one isn't, but that's because it's had a lot of chemical modification. Deoxyribose itself actually has 3 OH groups. Now, 2 of them have been used to make the connections to the linking phosphate groups. So there would have been an OH group here, and there would also have been one here. But you can see what's happened is that those 2 OHs have reacted with phosphate groups, what's called an esterification reaction, to make these links that hold the polymer together. The 3rd OH group would have been at this position, but you can see that those OHs have been substituted by these chemical groups here. Now, these are what biologists call the bases of the DNA. They're called bases simply because, as you can see, they contain groups like NH2 and NH here, which are basic. They can accept protons in the same way that ammonia does. As you can see, they're very similar to ammonia. But that's not actually important for their biological function in creating the DNA structure. So although we'll call them bases, we're not going to be focusing on that acid-base chemistry. We'll talk much more about these bases later, but the thing to notice for the moment is that there are 4 different ones here, and there are only 4 that you find in DNA molecules. So this one is called adenine, this one is called thymine, this one is called guanine, and this one is called cytosine. So we've got A, T, G, C, which if you've seen DNA sequences, you know it is the standard way to refer to the sequence of DNA, because that's the point, that they don't occur in this regularly repeating pattern in an actual DNA molecule. Instead, they occur in a particular sequence which encodes the amino acid sequences of the proteins that will be produced from the information stored in the DNA. So if we're going to understand how all this chemical functionality that you can see there comes together to form a 3-dimensional structure, we need to have a more realistic picture of what the molecule really looks like. Because you can see that some of the angles here, for example, this won't really be a right angle, as you know well enough from the work you've done this year. This is just a CH2 in the middle there, so it's going to be tetrahedral. So we need to have a more realistic picture of this molecule if we're going to start thinking about the intermolecular forces. So we'll go back to the molecular graphics software for that. So here we have a sequence again of 4 bases connected by this backbone running along here. So let's have first of all a look at the backbone. So you can see the sugar rings, so there's one, there's another one, and if we just look at those edge on, you can see here, for example, that they're not dead flat. They're actually puckered a little bit is the word that people use to mean that they're not absolutely flat. But you can also sense, I think, that there's not a lot of flexibility within those rings, simply because they're constrained by being, tightly coiled round into that 5-membered ring. So there's not much flexibility in the ring structures. But in between, so in between this ring here and this one here, you can count 1, 2, 3, 4, 5 single bonds. And one thing that you will have learnt this year is that when you have single bonds in a molecule, you can generally expect to have freedom of rotation around those. So in other words, this molecule doesn't have to have the bases arranged like this, because if you rotate around these bonds, you can make them point in all sorts of different directions. And what we need to think about is the implications of having this flexibility in the backbone here, which is what we would expect to have. On the one hand, of course, it's important because it needs to be there to enable the chain to fold around and create the double helix. If you didn't have that flexibility there, and it was fixed in some other structure, then obviously you'd have a problem. But on the other hand, the question is, if in principle it can be as flexible as having all those single bonds would make you think, how does it get pinned down into a single double helical structure? And that's what we need to really understand. How is that flexibility compensated for and effectively reduced to being a very limited flexibility by forming the double helical structure? Now, if we then turn our attention to the bases, we just rotate this around to look at these edge on, you can see something quite remarkable, which is that the bases are very different. They are absolutely dead flat. And there really is very, very limited flexibility there, because they really are constrained to be as flat as they look there. And so the question is why? And the answer, you've probably guessed already, because you've come across flat rings before, is that they are aromatic. So let's have a look at their chemical structures again and see how that can be. So here are the structures of the four bases that you get in DNA, and at first glance they don't look aromatic at all, in the sense that we've learnt it, because the only aromatic compounds that we've come across so far contained a benzene ring, which obviously is this, if you draw it out with the Kekulé formula, and we haven't got any of those kind of rings in these molecules. In part, that's because there are nitrogens all over the place. You can see these rings have a lot of nitrogens in them, and the first thing to say is that there's no reason why an aromatic ring should only have carbons in it. So there's no reason why you can't just replace one of those carbons with a nitrogen. So that compound, for example, is called pyridine, and it's a well-known aromatic compound. And so at a pinch you could see how that, for example, could be seen as an aromatic ring. But actually it's not as simple as that, because all of these are aromatic, and we saw that from their plenarity. So in order to understand how all of these rings are actually aromatic, we need to find a better way to explore how delocalization works in these structures. So let's start with the Kekulé structure for benzene, which is this. And obviously what we know is that that's not really a very good way of representing the structure, because you don't really have these three localized bonds. One way of thinking about that is that we could equally have drawn the Kekulé structure like this. So that instead of saying the double bonds are in those three positions, we could put them in the other three positions. And what you can now see is that the real distribution of electrons, which is the delocalized distribution around the ring, is a kind of average of those two. Okay, so in this case it's just an equally weighted average, where if we add up this and divide by 2, then in effect we get that, where each of the sides has, if you like, a half density pi bond, but in reality that just means continuous delocalization all the way around the ring. So organic chemists call these canonical forms, because what they're saying is you can't represent the true distribution of electrons by a single structure, but what you can do is draw a number of structures, in this case just 2, but it might be more than 2, and the real distribution of electrons will be some kind of average of those canonical forms. The other thing that's quite striking about this, and that turns out to be incredibly useful when you're thinking about delocalization, is that you can get from this structure to this structure with the help of curly arrows. Now you're used to using curly arrows to mean movement of a pair of electrons during a reaction mechanism, but actually what you can also do is think of it as, well, how do you get from one canonical form to another? The answer has to be by pushing pairs of electrons around. And obviously they don't really jump from one to another, it's just a way of exploring what the average will actually be. So if we push a curly arrow there, and then another one there, and then another one there, what you do is get from here to there. And so we can see that by pushing curly arrows we generate the alternative canonical, and then we say, right, the real electron distribution is some kind of average of those two. So we can apply that idea to some of these other structures. So let's start with cytosine as an example, because you can see it has 2 of the double bonds in place. So this half of the ring looks fine in terms of making it into an aromatic structure, but this one doesn't. This corner doesn't look right. But if we put that on there, because remember that nitrogen will have a lone pair of electrons because it's just got the 3 bonds, 2 to carbons and 1 to hydrogen, so it's got a lone pair left over. Then we can do the curly arrow trick and see how we can turn that into an aromatic structure just by doing this and that. And you can see that pair of electrons moving like that gives you an alternative resonance canonical, which is going to look like this. And again, what we're saying is that the real distribution of electrons is some kind of average of this and this. Now, it won't be equally weighted this time, because obviously these two have the same energy, because they're just different ways of distributing the electrons in exactly equivalent way. But here, you could imagine that this form's going to have a higher energy than this. It doesn't look as favourable having to separate the charges out like that. But nevertheless, this is going to make some contribution to the overall electron distribution, and you can see that this ring is indeed aromatic because you've got the 3 double bonds there, and so you could then push those 3 around and do this. And say, just like benzene, that you have this aromatic delocalization happening. Sorry, so that's going to be now like this. There we go. And we can do the same thing here. It turns out that 10 electrons delocalized around a ring also makes an aromatic system. So in fact, it's not just that these individual 6-membered rings are aromatic, but actually the whole system can have a 10 electron delocalized system running around the outside. So if we do the same as we did with cytosine and say, right, well, this group here, we could redraw that like this and say, right, that lone pair on the nitrogen can come into play to make a double bond there, and then that gives you O- out there. So if we now draw the electrons in that resonance form and then look at this side on and think about the p orbitals, then you can see how this is going to work. So if we start here, we've got a double bond there between carbon and nitrogen, so that what that actually means is that you've got 2 sideways overlapping p orbitals there. But we've also got a lone pair on this nitrogen here, so that can also be in a sideways overlapping p orbital. So that's 1, 2, 3, 4 electrons so far, because that one, remember, is a pair. So 1, 2, 3, 4 electrons so far. And we've got the double bond down here, so that's a pair of p orbitals there and there. 1 electron each, so that's 6 pi electrons so far. Then we've got a double bond between there and there, so that's a p orbital there and a p orbital there. Again, 1 electron each, so that takes you up to 8. And then we've got our double bond here, so there and there, 1 electron each, so that takes us up to 10. So we've got 10 pi electrons, and you can see there's continuous ring of pi electrons all the way around the outside of that system, and that's also an aromatic system. So these rings are not just partially aromatic, the whole thing is flat, the whole thing is aromatic, so that you can get that sideways overlap, and that's why you get those planar structures that we see in the DNA structure.